All right, hello everyone. We are here to work on the lipid and protein identification lab. And so I just wanted to show you exactly how that would have worked if we were in class. And so if you look at it, the purpose of this lab is to look at these 5 substances. I've got A, B, C, D, and E that they're labeled. And they're unknowns. And so your job would have been to use these different tests to figure out, first of all, is there a lipid in there? Is there a protein in there? Is there both, or is there neither one of them? So it's a possibility that it might not have anything, it might have both, it might have just one or the other. And so that's going to be the purpose of the lab. So your hypothesis is to guess which one you think of those 4 choices is in each of these substances. So you basically have 5 hypotheses, really. And for safety, we do have chemicals that we're using. I have Biuret's test, which is the one that's testing for proteins. And that's sort of a light blue solution. I don't know how well you can see that in there. But kind of a light blue solution. Sometimes people get it confused with Benedict's test because they are both blue to start off with. But that one doesn't require heat and it reacts with proteins, not carbohydrates. And then I also have, excuse me, I also have just a brown paper towel that I'm going to use for my lipid paper test. And remember, if it dries translucent when I put some of these materials on there, that means it's going to be a lipid. And if it dries back to normal, then it's going to be, there's going to be no lipid in it. Okay? And so safety-wise, I want to make sure that since I'm dealing with different chemicals that I'm trying to keep stuff off my skin and make sure I don't get it on my clothes because it could ruin my clothes. And I also want to make sure my eyes are covered, so I have my goggles again today. And so goggles are a must whenever we deal with anything with any sort of chemicals at all. And so the first thing we're going to do is go ahead and get our samples ready. So I have my well tray here. And my well tray is just that. It's this little tiny thing that has these little wells in it to hold small samples of materials that are liquid. And so I'm just going to take a few drops of each one, about 5 to 10 drops. Looks like it needs about 8 or so. And I'm just going to go straight across, make sure I keep them straight as to what they are, so they stay in the same order. And so I'll give you just a quick description on each of these, but substance A is kind of clear, but it has kind of a yellow tint to it. B has sort of some bubbles in it. C is a white substance, D is a clear substance, and E is kind of a cloudy substance. Okay? And so I just give you a little quick description on those. I'm going to move these out of the way so that we can sort of see the rest of the lab a little bit easier. So I'll slide these forward. And so the first thing I want to do is go ahead and do I'm going to set my fat test up. It takes about 10 minutes for these things to dry on a paper towel. And so we're not going to wait that 10 minutes. I did another one ahead of time to show you the results. But I'm just going to take a toothpick. Let me turn this around so you can sort of see that. But I've got the different labels, A, B, C, D, and E. And then I have a separate toothpick for each one just so I don't cross-contaminate anything. Okay, so I'm just going to take some of A. And I'm going to rub that on there. I'm going to get a different toothpick and take some of B. I'm going to rub that on there. A little bit more, make it so it's a nice mark. Some of C. Sometimes you need to get it a little bit more just so you can make sure you see it. And again, I'm just making a little wet mark on each one. And then some of it. And so hopefully, I don't know if you can see there's little wet marks on each one there for that. And so I'm going to put that off to the side, and we would normally just let that dry by itself. And then I would go ahead and get rid of my toothpicks because I really don't need those anymore. And now I'm going to go ahead and do my Biuret's test. My Biuret's turns sort of a lavender color or maybe a light pink color in the presence of a protein. So it says 5 to 10 drops. I'm going to add probably just a squirt here, which is about 10 drops for each one. And then I'm not sure how well you can see that, so let me kind of hold that up a little bit. I'm not really sure on the lighting there. But I'll put the results on the board for you there. I'm not sure if you can see the purple or not. But in terms of that, let me go ahead and just give you the results for the Biuret's test, because again, that one's pretty immediate. So biuretes, remember, either turns purple or there was no change. And so for substance A, there was no change. For B, it turned purple. For C, it also turned purple. For D, there was no change. And for E, there was no change. And then here is my sample of the different fat test results. And so you can see how B, D, and E dried kind of back to normal, but there's still kind of a wet mark on A and C. And so that's the translucence we were looking for there. So A was translucent. B was normal. C was also translucent. D was normal. And E was normal. Okay, and so from those results, you should be able to tell me and fill in the contents column as to what substances are in each of our unknowns. And so again, you have 4 choices. You can put lipid, you can put protein, you could put neither, or you can put both. And that's true for all of those. So your job is to figure out which ones are those are true. And then if you look at the lab sheet, there's 4 questions on the second page, and you're also writing a CER conclusion. Remember, if you need help on the formatting for CER, you can go to that lab report format sheet, which is one of those purple buttons on the home page. Again, if you need to rewatch this for more clarity, you can do that. That's the nice thing about videos, is you can always rewatch them and get more detail that you might have missed the first time. Other than that, you have a great day, and I hope you do well on your assignment, and I will see you next time.